Assalamu alaikum. My name is Dr. Aayesha Riaz. I'm an associate professor of microbiology in Agriculture University, Rawalpindi. I'm thankful to Third International Brain and Biomedicine Conference, IBBC 2026, to give me an opportunity to present my work in this prestigious conference. The topic of my talk is antibacterial activity of Korean red ginseng extract against E. coli isolated from the cases of bovine mastitis. Bovine mastitis, it is an inflammation of udder of ruminants and animals like cattle, buffalo, sheep, and goat, etc. And it is primarily caused by the pathogenic bacteria, including E. coli, Staphylococcus, Streptococcus, and many other. E. coli is the the largest cause of causing mastitis in animals, and it causes huge economic losses. E. coli, it is a gram-negative bacteria. It is an opportunist pathogen, as well as it's also a pathogenic bacteria for human beings, and it can be transmitted from milk to the human who consume the milk. It is the second most common pathogen isolated from mastitic milk samples, and according to a study, almost 46.18% is of prevalence is present in the dairy animals. Field surveys of major livestock diseases in Pakistan, they also have ranked mastitis as the number one disease of dairy animal because it causes the not only make the animal diseased but also decrease the quality of milk and reduces the production as well as cause severe economic losses. Therapeutic and subtherapeutic use of antibiotic as a result of mastitis can also increase the antimicrobial resistance in dairy animal because the only antibiotics they can cause give good treatment to the mastitis. But if animal develop antimicrobial resistance against those antibiotics, then they cannot be treated well, and that antimicrobial resistance can also be transferred to the human beings who consume the milk. So we were looking for some alternative therapy or some alternative cure for that in which there is no antimicrobial resistance is there. So we are thinking of using microbiological relevance of Korean red ginseng extract, which has so many properties, and it also has one of the property which is called as antimicrobial and immunostimulatory effect. Korean red ginseng contains bioactive ginsenosides and phenolic compounds with reportedly antimicrobial and immunostimulatory effects like anti-inflammatory. KRGE, which is Korean red ginseng extract, it has also shown inhibitory action against gram-negative bacteria. And while exploring this extract against E. coli isolates from mastitis provides insight into its potential mechanism of antimicrobial activity and efficacy against resistant pathogens. So our research aims to evaluate KRGE's antimicrobial potential as an alternative therapy for antibiotic-resistant E. coli. So the main objectives of our studies were to isolate, first of all, to isolate E. coli from milk samples obtained from animals which were infected by clinical and subclinical mastitis, and secondly, to determine the antimicrobial activity of Korean red ginseng extract against those isolated E. coli as an alternative therapy. So for this, we first of all collected few samples, include like 150 milk samples, which were positive for clinical or subclinical mastitis. Infected, and those samples were taken from cattle and buffalo. Our area of study was Rawalpindi district, and location of study was the bacteriology lab in Faculty of Veterinary and Animal Sciences at Agriculture University, Rawalpindi, to check that either clinical mastitis can be seen, whereas subclinical mastitis needs to be checked. So we checked that subclinical mastitis using the surf-field mastitis test, which is an on-farm test, and if it is positive, we to take the sample and bring it to the lab. So for other things, we first of all, when we come into the lab, bring the sample to the lab, we took the, first of all, we isolated the bacteria and purified that. Then also we biochemically identified that bacteria Also, we did some molecular biology research, which include DNA extraction and pathogenic gene identification, as well as antimicrobial resistant gene identification, because antimicrobial resistant genes, they are usually transferred to the other non-resistant bacteria, and they can get resistance, which is also called as the horizontal gene transfer. Then we perform antibiotic susceptibility testing on those and isolated bacteria to check that either they are resistant or not. And then we check the activity of Korean red ginseng extract on those isolated and resistant pathogens. So for in the results, we found out, first of all, we isolated the bacteria from the metastatic milk samples, and we first of all brought the samples into the lab and then grew onto the McConkie agar as well as EMB agar, which are specific for the E. coli. Then we morphologically identify those into the by using gram staining technique, and we found pink colored rods present in the under the microscope, which is also indication of the presence of E. coli. For biochemical identification, we did some API analysis in for the E. coli isolate, and here you can see that our samples have shown that they are gram negative E. coli. And in those tests included catalase test, methyl red test, citrate utilization test, and many other tests, which showed that our isolates are E. coli, and they are pure cultures of E. coli. Then from pure culture, we isolated the DNA, which we performed DNA extraction, and that DNA was used further into the multiplex PCR. Multiplex PCR, for that we used against three different pairs of primers. One is the STX1, sugar toxin producing genes, gene 1, forward and reverse primer. Here you can see that the amplicon size is 180 base pairs. And if you are using STX2, which is the sugar toxin producing gene 2, and it is also, if it is present, it will give an amplicon size of 255. And in the same way, if we use EAE gene, which is intimin gene, and it's also a pathogenic gene, if it is also present in the pathogen, so it will give a 384 base pair band. And these are the PCR conditions because we were using the multiplex PCR, and there were three pairs of primers. We used a annealing temperature 60 degrees centigrade. And second multiplex PCR which we used, it was against the antibiotics resistant genes, blatam and tetracyclines, and these genes, they were, if positive, it means that the bacteria has the resistant genes in them. The annealing temperature we used was 55 degrees centigrade. Here are the PCR results. In pathogenic genes, STX1, STX2, and EAE gene of bacteria, we found different results. Like for some 19 of the positive samples, they were positive for STX1 and EAE gene, 40% of the samples, and only 5 samples has 3 genes, which means these are highly pathogenic because they were having 3 pathogenic genes, and they were 10% of those. 9 of the samples, they had the STX2 and EAE gene, and 20% of those samples, and 13 samples, they showed only STX1, which is the 30%. So if you can see that these bacteria, they are different types pathogenic genes they have present in their genome. 46 of the E. coli sample, they were positive for these genes. Here you can see that the results of tetracycline and the other antibiotic resistant gene, and these were 57% of the samples were positive for the TEM gene, whereas the 43% samples were present for the tetracycline gene. We can see that a band here of 840 base pair for the TEM, and for TET A, there is a 590 base pair of the band. So the resistant pathogens, resistant isolates, were then used for the antibiotic susceptibility testing to confirm that. So we found out that our sample were also showing resistance. Here we can see the results that our samples were showing resistance against ampicillin, penicillin, and erythromycin, where there is a moderate or intermediate resistance against the gentamicin, ceftazine, and amoxicillin. Fluoroquinolones, they showed less resistant or more susceptibility towards these. So those isolates, they showed sensitivity toward these isolates. Now we used our resistant strains for the and we saw the results. We saw the activity of Korean red ginseng extract on these resistant pathogens. So we performed a method which is used, which is called as well diffusion method. The wells were produced into the agar, and then different concentrations of the Korean red ginseng extract was uh, uh, pour into those, including 50 microliter, 100 microliter, 200 microliter, and 400 microliter of the coriander ginseng extract was used. But these and then this agar, which is already had the lawn of resistant E. coli. And then we gave it almost 52, for 24 to 48 hours to show the activity. We found out that 60% were susceptible, while 40% were also resistant to Korean red ginseng extract. However, by increasing the concentration, the increase in the susceptibility and decrease in the resistance were seen, which means that the Korean red ginseng extract, it has the antimicrobial activity, but we need to calculate at which concentration it is showing the best activity. So, so at the end, I will conclude my talk that the Korean red ginseng extract was found to have antimicrobial activity against pathogenic resistant E. coli isolated from the patient's samples, and which also contain the resistant genes against which against resistant genes of the E. coli, which cause pathogenicity in the animals as well as in human beings. So, but Korean ginseng, as it is giving antimicrobial activity, its performance suggests potential as a supportive or complementary therapeutic option, which means that we can include this into our treatment along with the antibiotics, or as a supportive treatment, so that it can enhance the effect of antibiotics, or it can give a supportive effect for the treatment of the antibiotics. So there are some future recommendations that the future studies, we should also need to optimize the extract concentration and formulation to improve this antimicrobial activity, as well as further molecular and comparative analysis. They are also needed to explore synergistic effect and underlying mechanism of Korean red ginseng extract action. Also, field-based trials should be conducted to evaluate Korean red ginseng extract potential as an alternative or complementary therapy for bovine mastitis. So we can see that either it can treat the bovine mastitis or not. I'm really thankful again to IBBC and organizers of the IBBC too, which gave me an opportunity to present my work here. Thank you so much.